So hello students, welcome back again. Welcome back to Vedantu NEET English. And this is Baswaraj sir, your biology master teacher. Yes, there was some issue with the stream. I think the issue has been resolved right now. So students, let me know in the chat if I am audible and visible to every single one of you. Quickly let me know in the chat if I am audible and visible to all of you. Even the presentation behind me is visible to all of you. The structure of a cell. Is it visible to all of you? Quickly let me know in the chat if I am audible and visible to all of you. Yes, I think I'm visible, right? I think I'm audible and visible to all of you. Now students, let's quickly recap what we did in previous class, because some of you will be missing it in recorded session, right? So in previous class, I'll ask you a lot of questions now. Quickly answer. In previous class, we spoke about prokaryotic cell. Now tell me, the prokaryotic cell, is it membrane bound or is it does not have any membrane? Quickly let me know in the chat. Do you think does it have a membrane or does it not have a membrane at all? Obviously students, your prokaryotic cell, the organelles are without a membrane. Now a prokaryotic cell has three different layers. The outer layer is called as glycocalyx. Glycocalyx can be of two types. It can be made up of tough and thick sheets or loose sheets. Tough and thick sheets called as capsule, loose sheet called as slime layer. Then we have second layer called as cell wall, which is made up of peptidoglycan. Then we have the innermost layer, that is the plasma membrane, which is basically selectively permeable in nature. And we also have a 70S ribosome. We also have DNA. We also have the genetic material. Students, am I visible and audible? Yes. Beautiful, no? Yes, I'm audible and visible to all of you. Now students, with that being said, all of you should know the eukaryotic cell. All of you should know eukaryotic cell is all the organelles present in the cytoplasm. All the organelles in the cytoplasm are membrane bound. They're going to be membrane bound. All of you know that. They're going to be membrane bound. Students, quickly mark your attendance. If you're watching it live or watching it recorded, quickly mark your attendance and smash that like button. No, Hulk smash it. And punch you give it. Nicely punch you give it and you will understand. Okay. Now, eukaryotic cell includes all protist, plants, animals as well as fungi. Now tell me, which kingdom is missing from here? I already told you. Which kingdom is missing from here? The kingdom which is missing from here is kingdom Monera. Kingdom Monera is missing. Kingdom Monera is missing. And kingdom Monera, if it does not have eukaryotic cell, so kingdom Monera will be prokaryotic in nature. It will be prokaryotic cell. Prokaryotic cell in kingdom Monera. Apart from kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, kingdom Animalia, all of them have eukaryotic cell. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm. That is, within the cytoplasm, there are many different compartments. Through the presence of membrane-bound organelles. Eukaryotic cell possesses an organized nucleus with a nuclear envelope. See, in the case of prokaryotic cell, the nuclear envelope was absent. Now here there is nuclear envelope is present here. Right? In addition, eukaryotic cell have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. Tell me, is the chromosomes condensed or decondensed? Obviously, chromosomes are completely condensed. Okay. Now, all eukaryotic cells are not identical. Obviously, plant cell and animal cell are totally different. Plant cell is different, animal cell is different. Quickly, write down the difference now. What is the difference between a typical plant cell as well as a typical animal cell? What is the difference now? Typical plant cell and typical animal cell. Now, students, the first main point, cell wall. Do you find cell wall in the case of plant cells? Yes or no in the chat quickly. Do you find cell wall in the case of plant cells? Yes. So cell wall is present here. Here we have cell wall is present. Okay. What about animal cell? The cell wall is going to be absent. In the case of your animal cell. Now, what about plastids? Do you find plastid in the case of plants? True or false? Quickly. In the case of, do you find plastids? Do you find plastids in the case of plants? Yes. plastids are present. Do you find plastids in the case of animals? No. Plastids are not found in the case of animals at all. Okay, plastids are not found. Now what about large central vacuole? A large central vacuole. Large central vacuole is present in the case of your plant cell. Now students, tell me, do you find vacuoles in the case of animal cell? Quickly go. True or false? Do you find vacuoles? Do you find vacuoles when it comes to animal cell? Quickly. True or false? Do you find vacuoles? Quickly. Go. Obviously, you find vacuoles. You will find vacuoles, but they will not be large. They'll be tiny. That is the only difference. So you will find vacuoles in the animal cell, but they are tiny. That's all. It is true. You have vacuoles in the case of animal cell, but they are tiny. Remember that. Okay. Remember that, all of you. You find vacuoles. Now, apart from that, there is one structure which is present only in animal cell and absent in plants. Which is that? Tell me. Tell me which is that. You find one organelle. It is present in the case of animal cell, but absent in the case of plant cell. Centrioles. Yes, centrioles. Centrioles are present in the case of animals, but they are absent in the case of plant cell. They are absent in the case of plant cell. Done? Done? Yes. Now students, now we'll go here, we'll read here. Plant and animal cells are different as former per cell wall. That is, plant cell has cell wall, plastid and large central vacuole, which is absent in the case of animal cell. See, large vacuole is absent, but vacuoles are present. Vacuoles are present. On the other hand, animal cells have a centrioles, which is absent in the case of almost all plants. Done? Any doubt in paragraph 1? Students, tell me quickly in the chat. Any doubt in paragraph 1? No doubt. No doubt at all. Nice. Now, next paragraph. Next paragraph. Now, this is your, tell me, which one is an animal cell here? Tell me quickly in the chat. Which one is an animal cell? Which one is a plant cell? Tell me. Tell me, on my right side, what is this? On my left side, what is this? Students, quickly look at the vacuole. Where do you find large central vacuole? Quickly tell me. Here do you see a large central vacuole? Yes, this is a plant cell. In the case of animal cell, do you find any type of large central vacuole? No. So large central vacuole is absent. But here we have centrioles in the case of animal cell. Here in the case of plant cell, we find cell wall. We find cell wall. We also find plastids. Where is the plastids? Plastids, chloroplast here. Plastids are found here. Plastids are found here. So that is the difference between your plant cell as well as animal cell. Easy. Now students, can we understand cell membrane now? Students, I believe it's visible, right, to all of you? Quickly tell me in the chat, is it visible? Because I made sure the entire paragraph is at one point, because it is very important to understand every single line here. Question can be asked from any single line here, every single line. Visible? Visible, right? All of you quickly tell me in the chat. Visible, visible, yes or no? Visible? Yes, visible. Now students, I want you to open your NCERT along with you. Keep your NCERT next to you. Read the lines with me. Understand with me. Okay. The detailed structure of membrane, cell membrane, was studied only after the advent, that is, advent means discovery of electron microscope. Why? Because if you do not have electron microscope, you cannot study the detailed structure of membrane, cell membrane. To study the detailed structure of cell membrane, we need the electron microscope. Okay. In 1950s. Meanwhile, chemical studies on cell membrane, especially human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. So even without the electron microscope, some amount of studies were done. See, they didn't wait until electron microscope. Before the discovery of electron microscope, some studies were done on the cell membrane, especially on RBC. Why? Why do you think? Why do you think RBC? Because students, remember, RBC is empty from the inside. It doesn't have any major organelles. Mostly it is made up of hemoglobin. right? So it becomes a very ideal test subject. So all the scientists, majority of work was done on RBC. Most of the work was done on RBC. Okay. Now, these studies showed that cell membrane is mainly composed of lipids and proteins. So if someone asks you, what is cell membrane made up of? What is cell membrane made up of? What are the main components of cell membrane? You should tell them, sir, cell membrane is mainly made up of lipids as well as proteins. Lipids as well as proteins. Lipids as well as proteins. Okay? Lipids as well as proteins. Next. Okay? The major lipid are phospholipid. So that is also very important. Which type of lipid is there? If you talk about, major component of lipid is going to be phospholipid. It's going to be phospholipids. Phospholipids are main component of cell membrane. Phospholipids. Okay? Phospholipids that are arranged in bilayer. So phospholipids are arranged in a bilayer. What is this bilayer, students? Let me draw it for you. This is the bilayer, students. This is the bilayer. Can you see? This is going to be the bilayer. This is going to be the bilayer. This is the bilayer, student. This is the bilayer. Okay. Bilayer. Also, lipids are arranged within the membrane with a polar head towards the outside. Okay. I think I did the ulta. Ulta we did, no? Ulta we did. Yes. 1 second. This was wondering, why does it look so weird? Okay. Saturated fatty tail. This is important. This is nice now. This is nice. Okay. The lipids are arranged in the membrane within the polar head towards the outside. This is going to be the polar head. This is going to be the polar head. Okay. And hydrophobic tail. Can you see this was the non-polar tail, which is this non-polar tail. Students, this non-polar tail is going to be hydrophobic in nature. It's going to be hydrophobic in nature. Hydrophobic in nature. That is, it doesn't like water. Okay. It doesn't like water. Now this is polar head. Polar head is hydrophilic. It's going to be hydrophilic in nature. It loves water. Okay. Polar head. Okay. Polar head is going to be hydrophilic in nature. This is going to be hydrophobic in nature. That is the tail. Okay. That is the tail. Nice. Okay. Hydrophobic tail towards the inner part. Now this ensures that the non-polar tail, see, the non-polar tail of the, see, very important line, non-polar tail of saturated hydrocarbons. So if you get a question in examination, what is the tail made up of? What should be your answer? Tell me. What should be your answer? If they give you a question in the examination, what is this tail made up of? Saturated fatty acids or unsaturated fatty acids? What should be your answer? Quickly tell me in the chat. It is supposed to be saturated conditions. Saturated hydrocarbons. Saturated hydrocarbons, that is single chain, no double chain, single chain, single bond, right? Is protected from the aqueous environment. That is, what do you mean by aqueous environment? Here we have water everywhere. Here we have the water everywhere. Aqueous condition. Okay. Aqueous condition. Clear. Okay. Now, in addition to phospholipid, membrane also contains cholesterol. Now again, you should remember, it will have cholesterol, but why? Why will there be cholesterol? What is the requirement of cholesterol here? The answer is very simple. It to make the membrane fluid. Students, remember, this is a membrane, and this membrane is extremely fluid in nature. It is extremely fluid. It's something called as quasi-fluid in nature. And meaning of quasi-fluid is semi-fluid also. And the fluidity of membrane is given by cholesterol. It is given by cholesterol. Okay. Now, later, biochemical investigations clearly revealed that the cell membrane also possesses protein and carbohydrates. It also has protein as well as carbohydrates are also present here. So carbohydrates are also present. So what is cell membrane made up of? Lipids, carbohydrates and protein. Which type of lipid? Phospholipid. How is it arranged? Polar head towards the outside and non-polar tail towards the inside. Polar head towards the outside, which is going to be completely hydrophilic in nature, and non-polar tail towards the inside, which is going to be hydrophobic in nature. It is going to be hydrophobic in nature. Clear? Clear, students? Now, students, the ratio of protein and lipid varies considerably in different cell type. For example, heart cell, it will have more heart cell, cardiac cells, it will have more protein and less fat. The cheek cells will have more fat and less protein. Do you get it? That is going to be the difference. That is going to be the difference here. Okay. See, in human beings, in the case of human beings, the membrane of erythrocyte, that is RBC cells, red blood cells, erythrocytes, has approximate 52% protein and 40% lipid. 40% lipid and 52% protein. Points are very important. Each and every point is very important. Depending upon the ease of extraction, again, very important line, students. Here, I told there are proteins. Here I told you there are proteins inside. The proteins can be of two types. The proteins can be of two types mainly. Some proteins can be easily extracted. They can be easily removed. Some proteins are very difficult to remove. Now you tell me quickly. Imagine I have a. Now imagine this is a ground. This is a ground. Okay. Now if I keep this pen almost towards the inside, if the pen is towards completely inside, is it difficult to remove the pen or easy to remove the pen? Tell me. Obviously, if the pen is all completely inside, difficult to remove the pen. Yeah, difficult to remove the pen. Now imagine if the pen is almost here. Tell me now, is it easy to remove the pen or difficult now? Now it is very easy to remove the pen. Now that is called as ease of extraction. That is called as ease of extraction. Sometimes what happens is, some proteins are completely inside the membrane. They are completely inside the membrane. Such type of proteins are called as integral proteins, which are completely inside or partially inside. Completely inside or partially inside the membrane. Some proteins are present towards the outside, like this. Can you see? Some proteins are present towards the outside completely. Such proteins which are present on the outside is called as peripheral proteins, which are very easy to remove. So based on the ease of extraction, there are two types of proteins. Ease of extraction, there are membrane proteins, can be classified into integral and peripheral. Peripheral proteins lie on the surface. They are on the surface, easy to remove, lie on the surface of the membrane, while integral proteins, one which are inside, proteins are partially or totally buried in the membrane. They are completely buried inside the membrane. So two types of proteins, peripheral as well as integral proteins. Any doubt there? Quickly ask me. Any doubt there? Yes? Easy? Difficult concept? Tell me, did you understand the difference between the structure of plasma membrane? Polar head towards the outside, non-polar tail towards the inside. Yes. Lipid, phospholipid, proteins, two types, and carbohydrate is also present. Carbohydrate is also present here. Okay. Carbohydrate is also present here. Now, next concept. This is the entire structure. Can you see? This is going to be a plasma membrane. See, polar head towards the outside, non-polar tail towards the inside. This is phospholipid bilayer. There is peripheral proteins on the outside, and there is also proteins on the inside. That is integral protein. This is sugar, basically your carbohydrates. Sugar, which is carbohydrates, are also present. Then we have cholesterol here, which provides fluidity. Cholesterol. Done. Any point missing? No point is missing here. No point is missing. Now, students, now there is a model given. There is a model given. Yes, there is a model. There is a beautiful model here. What is the model here? An improved model of the structure of cell membrane was given by two scientists, Singer and Nicholson. Singer and Nicholson in the year 1972. Singer and Nicholson gave a cell membrane model in the year 1972. 1950 microscope, 1972 Singer and Nicholson model. Dates are very important. Widely accepted as fluid mosaic model. Now, what is the meaning of fluid mosaic? Mosaic means asymmetry. Mosaic means asymmetry. Now, doesn't it look like a road? You're nicely going in your bike, one hump will come, one more hump will come. Again you're going, one more big hump will come, one more hump, one more depression, hump, depression, hump. So it is like an Indian road. This is like an Indian road. This is called as mosaic asymmetrical mosaic condition. It has complete mixture of carbohydrates, proteins, lipids, completely mosaic in nature. Okay. Now, mosaic model. According to this, the quasi-fluid. The meaning of quasi-fluid. Semi-fluid. Semi-fluid of lipid enables lateral movement. I'll tell you, students. Understand the movement now. Understand the movement. There are two types of movements here. There are two types of movements here. One is called as lateral movement. Other one is called as flip-flop movement. Okay, flip-flop movement. Lateral movement is mostly shown by lipids as well as some proteins. Okay, lateral movements, and this happens in the monolayer. Let me tell you, flip-flop movement is only shown by lipids. It happens between the layers. Understand now. This is the polar head. Okay, polar head. This is the polar head. Okay, this is the non-polar tail. Polar head as well as non-polar tail. Now, what is lateral movement now? Understand the lateral movement now. Very simple concept. Understand the lateral movement concept. In the case of lateral movement, what happens is, in the case of lateral movement, this will go here, or this can come here. Basically, the movement of lipids laterally. That is called as lateral movements. And this is happening in a monolayer. Only this layer is involved. See, there are two layers, right? This one layer is involved in lateral movement. Lateral movement will never happen between the layers. It will happen within the layer. So this will go here, this can go here, or this can go here, or even this can even go here. Mostly it is going to be not at very far, doesn't happen. Basically this will go here and this will go here. That is called as lateral movement. Laterally they are moving. Okay. Even the proteins can show it. Even sometimes proteins can move laterally a little bit. Proteins can move laterally. Okay. Now what is flip-flop movement? Flip-flop movement is very simple. Understand. What happens in flip-flop movement? In the flip-flop movement, this particular polar head will go this side and this polar head will go this side. That's why it is between the layers. So this is called as flip-flop movement. So this particular polar head will move this side and this polar head will move towards the other side. Flip-flop movement. Flip-flop movement. Understood? This is lateral movement. This is lateral movement. This is lateral movement. This is flip-flop movement. Flip-flop movement. Understood? Yes. Beautiful. Easy to understand. Quasi-fluid nature, and because all this movement, all of this movement is because the membrane is semi-fluid in nature. Membrane is semi-fluid in nature. That is why all of this is possible. Lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. Okay. Now, the fluid nature of membrane is also important from the point. This is the functions. Some of the functions are given here. Functions. Okay. The fluid nature of membrane is also important from the point of view of functions like cell growth. Obviously, if the cell has to grow, the cell plasma membrane has to expand a little. Yes. Formation of intercellular junctions, that is connection between the two cells, the cytoplasmic connections, plasmodesmatal connections. Yes. Secretions, endocytosis and cell division. All of these are functions of cell membrane. Functions of cell membrane. Clear, all of you? Clear? Tell me. Clear? This paragraph is clear to all of you? And with that, we have one more last paragraph. Main function. This also is this one right here is also a main function. Main function again. Main function is given here. One of the most important functions of plasma membrane is the transport of molecules across it. Obviously, plasma membrane can make sure some molecules are entering and some molecules are exiting. Yes. But tell me in the chat, will it allow the complete movement of molecules? Tell me. Will it allow the complete movement of molecules? You're like, no, you go inside, I'll come outside. No. There is always some form of restriction. Yes, there is some form of restriction is there. Right? The membrane is selectively permeable. See, this is the restriction. Selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this type of movement, when there is no energy requirement, that is called as passive transport. No energy is required, molecules are passing through. Passive movement. Neutral molecules may move across the membrane by process of simple diffusion. By simple diffusion, all the molecules, neutral molecules can pass through. They can go and come back. Okay. Along the concentration gradient, that is from higher concentration to lower concentration they can go. Higher concentration to lower concentration, without any difficulties, neutral molecules can pass through. Okay. Now again, water can do the same thing. Yes, water can do the same thing, and that is called as osmosis. That is called as osmosis. Okay. As polar molecules cannot pass through the non-polar lipid bilayer. Polar molecules cannot pass through the non-polar lipid bilayer. They cannot pass through. Polar molecules cannot pass through. Okay. They require a carrier protein. So if they cannot pass through on their own, they require a carrier molecule. They require a carrier. The carrier is a protein. Okay. They require a carrier protein of the membrane to facilitate their transport across the membrane. Simple definition, students. Imagine you're going from your house to your friend's house. You're going from your house to your friend's house and you cannot walk and go. What do? You call your other friend, like, Macha, bring one bike, you come, you pick me up, and then we'll go together. That is what is happening here. Since the non-polar molecules cannot go past through the barrier, they're taking the help of a protein. Now, protein will take the molecule and make it cross. Protein will take the molecule and make it cross now. Okay, make it cross. A few ions or molecules are transported across the membrane against their concentration gradient. That is from now against the gradient now, from lower concentration to higher concentration. Lower concentration to higher concentration. Such transport requires energy because see, you are going from your house to your friend's house. You need you need petrol. Petrol is required. No, energy is required. See, energy is there. See, lower concentration to higher concentration. Such transport is an energy dependent process in which ATP is utilized, and it's called as active transport. Very simple. Active transport requires energy. Passive transport doesn't require energy. Passive transport, what happens? From higher concentration, lower concentration. Active transport from lower concentration to higher concentration. Lower concentration to higher concentration. Done. Very simple, no doubt should be there. Very simple, no doubt should be there. Done. Example is sodium potassium pump. Okay. Now students, with that your first part of your today's class is over. Now we'll go to the cell wall now. See, first layer is always not the first layer. This cell wall is obviously outside, but plasma membrane is the major chunk. It is over. Now we go to the cell wall now. Now we go to the cell wall. Okay, let's read it. As you may recall, all of you should recall now. All of you should recall. A non-living rigid structure is called as cell wall. So if I ask you true or false, cell wall is living or dead? Answer go in the chat quickly. Cell wall is living or dead? What is the answer? Quickly in the chat. You will tell me, sir, cell wall is dead. Plasma membrane was living. Plasma membrane was living. Okay. It forms the outer layer and outer covering of the plasma membrane. It makes the outer covering of the plasma membrane of fungus and plants. Fungus and plants. Now tell me students, what about algae? Do you think algae has the cell wall? Yes or no in the chat quickly. Do you think algae has cell wall? Yes or no? Quickly. Quickly in the chat. The algae, does it have cell wall? Yes sir, algae also has cell wall. Why? Because algae is also a plant. Algae is also a plant. Remember that, okay? Now, the cell wall not only gives the shape, see function again, this is the function. Function. The cell wall not only gives the shape to the cell and protects the cell. See, first one is here. Shape of the cell is first one. Second one is protection. Protects the cell from mechanical damage or infection. It also helps in third one here. Cell to cell interaction, and fourth one, produces barrier to undesirable macromolecules. 4 functions is right here. Done? Done. Now students, algae, very important. Write down this. What is algae cell wall made up of? Algae cell wall is made up of what? Algae cell wall is made up of your cellulose plus galactans, given red, galactans, mannans, galactans as well as mannans, plus minerals like calcium carbonate plus calcium carbonate. This is what algae cell wall is made up of. Cellulose, galactans, mannans, calcium carbonate. Now what about plant cell wall? What about plant cell wall made up of? Okay, let's go. Now, in other hand, plant consists of cellulose, hemicellulose, pectins and proteins. Now what about fungi? Quickly tell me in the chat. What about fungi? What is the cell wall of fungi made up of? It is not given here, but I'm asking you. Fungi is made up of chitin. It's made up of chitin. Remember that. It is made up of chitin. Fungi is made up of chitin. Okay, it is made up of chitin. Okay. Now students, the cell wall of young plant is called as the primary wall. It is called as primary cell wall. Understand here. Here we initially in the case of plants, initially in the young plants, there is something called as the outside wall. Can you see? initially in the case of young plants, there is something called as, can you see the outside one? That is called as the primary cell wall. It's the primary cell wall. Primary cell wall is found in the case of your young plants. Primary cell wall is found in the case of young plants. Okay, young plants. And this primary cell wall is mainly responsible for the growth, and it is mostly permeable. It is mostly permeable in nature. Primary cell wall. Now students, within the primary cell wall from the inside we have the secondary cell wall now. Can you see? Within the primary cell wall from the inside secondary cell wall is growing. This is the inside one, is called as the secondary cell wall. Secondary cell wall grows inside of primary cell wall. It grows inside the primary cell wall. So it's on the inner side. Inner cell side. So now students, this primary cell wall is also elastic in nature. It is also little elastic in nature. That's why it is able to grow. But your inner cell wall, students, it is non-elastic in nature. Non-elastic in nature completely. Why? It is non-elastic in nature because it has depositions of lignin. It has depositions of lignin and suberin. Lignin and suberin depositions are there. That's why it is non-elastic in nature. It is non-elastic in nature. Now students, the primary cell wall which is present on the outside is permeable, but your secondary cell wall is going to be completely non-permeable or impermeable. It's going to be completely impermeable. Clear, all of you? Clear? So we have primary cell wall present in the young plants, which is replaced by primary cell wall is completely replaced by secondary cell wall. It is replaced by secondary cell wall. Primary cell wall is young plants, growth, permeable, elastic. Secondary cell wall is inner side, non-elastic. Why? Because lignin and suberin depositions, and it is completely impermeable in nature. Okay. See here. The cell wall of young plants, the primary cell wall, is capable of growth, which gradually diminishes. I told you, the primary cell wall on the outside gradually diminishes as the cell matures, and secondary cell wall is formed on the inside, on the inner side towards the membrane side of the cell. Now students, now this was one cell, students. This was one cell. Imagine there are multiple cells here. This is one cell wall. Okay. Now we have one more cell here. I hope this is visible. I hope this is visible to all of you. Okay. Now students, there are two cells here. I don't know, one is bigger. Now, imagine there are two cell wall here. Between the two cell walls of a plant or fungi, here, between the two cell walls, there is a small structure here. Between the two cell walls, there is a small structure called as middle lamella. This is called as middle lamella is present. Now tell me students, this is the cell wall outside. Inside the cell wall we have plasma membrane. Yes, inside the cell wall we have the plasma membrane. Yes, inside the cell wall we have the plasma membrane. Yes, we have the plasma membrane. Now students, between the two plasma membranes there is some connection that is called as plasmodesmata connection. This is plasmodesmata connection. The two plasma membrane are attached here. Now what is middle lamella? Middle lamella is present between the two cells, between the two cell wall. Actually this is the cell wall. Outer one is called as cell wall, the inner one is called as plasma membrane. Between the two plasma membrane, plasmodesmatal connection. Plasmodesmatal connection between the two plasma membranes, cell to cell connection. Actually it is actually the cytoplasmic connection. The entire cytoplasm connected. Entire cytoplasm is connected here. Between the two cell wall there is middle lamella. There is middle lamella. Understood all of you? Between the two cell wall there is middle lamella. Between the two cytoplasms, connecting the two cytoplasms, we have the cytoplasmic connections. We have the cytoplasmic connections. Okay. See here. Now, the middle lamella is a layer mainly of calcium pectate. Also very important. It is made up of calcium pectate which holds or glues the different neighboring cells together. So it is a glue which is present between them. Made of calcium pectate. Made of calcium pectate. Here. Calcium pectate. It is made up of calcium pectate, middle lamella. Okay. The cell wall and middle lamella may be transversed by a plasmodesmata. I told you. Transversed by plasmodesmata which connects the cytoplasm of neighboring cell. This is the plasmodesmata. Plasmodesmatal connections we tell, right? Plasmodesmatal connections. This is plasmodesmata. Plasmodesmata. Plasmodesmata, which is connecting the cytoplasm. This is the cytoplasm, right? This is the cytoplasm. It is connecting the two cytoplasms. Connecting the two cytoplasms. Done? See here. This is the plasmodesmata. The two cytoplasm connected. This middle part, that is middle lamellae. Here it is middle lamellae. This is plasmodesmata. That is the, it connects the two cytoplasms of two different cells. Understood? Next. Students, with that your cell wall is over. With that cell wall is over. Done. Difference between, you should know the difference between middle lamella and plasmodesmata. Okay, know the difference between them. Middle lamella is present between the two cells, it's like a glue, that's all. And there is something called as transverse section which is connecting the two cytoplasms. Between the two cytoplasm connection is called as plasmodesmata. Nice. This is the last line, very important. Plasmodesmata. Now, can we start endomembrane now, students? Can we start now endomembrane? Yes. Now we will start the endomembrane system. Students, if you are able to understand what I am teaching, right? Quickly, I want everyone to comment in today's video how was the class, and also smash the like button. Quickly smash the like button, all of you. Mark your attendance and let me know in the comment section how was today's class. And I've been reading your comments. And Ravindrani, don't worry, I am researching. It's very difficult to get information on other states. Ravindrani, I'm searching for the dental colleges and everything. I will get to you. The Ayush counseling, right? Don't worry. Okay. Now, endomembrane system. We'll start with endomembrane system now. Now, endomembrane system is like one small close friends group. Tell me, all of your friends group in the class, you have some best friends, all of you will make a small group in the class. Now tell me, why do you have a small group of friends? Because all of you will have a similar interest. All of you will love anime, one anime group. All of you will love songs group. All of you love bikes, one biker gang will be there in the class. It will be there, no. Some of you like sports, one sports group is there, right? So based on the interest, there is some type of group formation in the classroom? Yes. Similarly, inside your cell, similarly inside the cell also there is a small group. The main reason for this group is because all the functions are coordinated here. All the functions are coordinated. Like I have the same function, you also have same function. Functions can be completely not same function, but they're coordinated with each other. Okay, they're coordinated with each other. Someone is saying, sir, I am alone, lone wolf. Huh? Song group, I am myself. Don't need anyone in life. I am all alone. Nice, thug life, thug life. Okay, understand now. Now, while each membrane organelle is distinct in terms of structure and function, many of these considered together as endomembrane system because their functions are coordinated. Because some of these organelles functions are coordinated, we call them endomembrane. Endomembrane. Okay. The endomembrane system includes endoplasmic reticulum. Do you want a small trick here, students? Do you want a small trick here? The small trick is every gamer loves victory. Every gamer loves victory. E for endoplasmic reticulum, G for Golgi complex, L for lysosomes. Where is it? Lysosomes. Lysosomes. Yes. L for lysosomes and V for vacuoles. V for vacuoles. V for vacuoles. Every gamer loves victory. All of you type this in the chat and remember it also. Remember it. So all the 4 organelles are endomembrane, part of endomembrane. Why? Because their functions are coordinated. Okay. Since the functions of these organelles, function of mitochondria, chloroplast and peroxisomes are not coordinated with the above components, these are not considered part of endomembrane system. Students, they'll give you question. They will give you just simple question. They'll give you 4 options. I will only give you, tell me the answer. Which of the following is not a member of endomembrane? Which is not a part of endomembrane? First option is ER, second option is Golgi complex, third option is vacuole, fourth option is peroxisome. Tell me the answer now. Fourth option is peroxisome. Which of the following is not a part of endomembrane? NEET question has come like this. Such easy questions will come. It's only a matter of how you're prepared for it. Tell me the answer now. Tell me the answer. Which organelle is not a part of endomembrane? Option number 1 is endoplasmic reticulum. Option number 2 is Golgi complex. Option number 3 is lysosomes. Option number 4 is peroxisomes. Obviously peroxisomes then. Peroxisomes. So you will get such questions. Okay. Endomembrane. Now students, can we start with endomembrane? The first one now. First part of your endomembrane. Very simple. Now tell me, what is the meaning of endo? What is the meaning of endo? Endo means inside. Endo means inside. Okay, inside. Plasmic means what? Cytoplasm. Right? Reticulum means what? Reticulum means network. So inside the cytoplasm, there is some form of network. That is nothing but endoplasmic reticulum. That's all. So inside the cytoplasm, there is a network formation. Okay. Now listen to me very carefully. Imagine this is a cell. This is a cell right here. Okay, this is a cell right here. We have a cell. Here in the middle, what do we have, students, in the cell? The middle part is called as the nucleus. We have the nucleus here. Okay. Then we have the nuclear envelope. Yes, this is the nuclear envelope. Okay, nuclear membrane. Let's say nuclear membrane. nuclear membrane. Now students, understand, from this nuclear membrane, from the outer side of nuclear membrane, there is the development of ER. So if I ask you from where is the endoplasmic reticulum developing? Endoplasmic reticulum is developing from the outer membrane of nucleus. Outer membrane of nucleus. This is your ER. This is your endoplasmic reticulum. Endoplasmic reticulum is developing from the outer layer of nuclear membrane. Now students, this endoplasmic reticulum is dividing the cell into two parts. This endoplasmic reticulum is dividing the cells into two halves. Whatever is present inside the ER here, whatever is present inside the ER, we call that luminal. Luminal. Okay. Whatever is present inside the ER, that part of the cell, that part of the entire cell is called as luminal. That is called as luminal. Now, what is the present outside? Whatever is present here, can you see this part? Whatever is present on the outside, basically the entire cytoplasm, that is called as extraluminal. Extraluminal. Clear? So it is dividing the entire cell into two parts. Whatever is present on the inside is called as luminal. The outside part is called as extraluminal. Okay. Let's read now. Okay. Endoplasmic electron microscope studies of eukaryotic cell reveals the presence of network. See, I told you, presence of network or reticulum of tiny tubular structures. Network of tiny tubular structures scattered in the cytoplasm is called as endoplasmic reticulum. It is called as endoplasmic reticulum. Okay. Now, hence ER divides the intracellular space into two distinct compartments. Luminal, inside the ER is called as luminal, and extraluminal, that is basically the cytoplasm. So extraluminal can also be called as cytoplasm. Same meaning. Extraluminal, the same meaning is cytoplasm. Students, every line of NCERT is being covered. Every single line we are covering. We are not missing anything here. If you do this, trust me students, if you do the entire, if you religiously, properly finish the entire series of line by line, you will be so confident with your NCERT. Any part of the question you can solve. Any question you can solve. That much of confidence you need. Okay, and we'll make sure here you get the same confidence by solving every single point to point of your NCERT. Okay, point to point of NCERT. So when you do this and when you read this on your own, it becomes so easy for you also. I know this will take more time. Obviously, this will take more time. I am teaching you every single line of NCERT. I am drawing and teaching you line of NCERT. This will take more time, but that effort is counted. That effort means something, right? So all of you who are being regular, see, some students will come and some students will go. But if you are regularly attending such classes and understanding every point of NCERT, you are good to go. You're good to go. Now students, the ER often shows ribosomes. Tell me, ATS ribosome, it's eukaryote. ATS ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes. So if we have ER plus ribosomes, such endoplasmic reticulum with ER is called as RER, rough endoplasmic reticulum. It's called as rough endoplasmic reticulum. Yes, rough endoplasmic reticulum. Thumbnail I have to change. I am not, see students, I am very bad taking pictures. I am very, very bad taking pictures. So I will change thumbnail. What is wrong with thumbnail? Tell me. What is wrong with thumbnail? What is wrong with thumbnail? Is there any mistake? I have to check. The photo, no. Oh God. Yeah. Diksha ma'am name is there. Oh, I'll get it changed. Don't worry. I'll get it changed. So my name is Diksha ma'am from today. Okay. I will get it changed. Don't worry. I will get the photo and the name changed both. I'll get it. Don't worry. Don't worry. I'll get it changed. Don't worry. Okay, I'll get it changed. Don't worry. Or my name is Diksha ma'am from today. Who cares? Only thing which matters is what you're studying here. Okay. Write your name. Yeah, I'll write my name. Don't worry. All of you know my name. No. All of you know my name. So what is there? Chill. Okay. The ER often shows. Okay, done. Right. Rough endoplasmic reticulum. In absence of ARIA ribosomes, they appear to be smooth. So ER plus no ribosomes plus nothing is called as smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Done. Now RER function is very important. Function is very important. Okay. RER is frequently observed in the cells actively involved in protein synthesis and secretion. So students, tell me, what is the function? Hi. Hello. My name is Diksha ma'am. Hello. Welcome to you. I'm your chemistry master teacher. Students, beta understand. Okay. So your rough endoplasmic reticulum main function is going to be protein synthesis. Now, where will the question come from? Beta, the question will come from the smooth endoplasmic reticulum, because the main function of smooth endoplasmic reticulum is releasing of lipids and steroidal hormones as well as steroidal hormones. Okay, beta. So they are extensive and continuous with the outer membrane. See, we did this line. I told this line. Rough endoplasmic reticulum is extensive of the nuclear membrane. Extensive of nuclear membrane. Okay. The smooth endoplasmic reticulum is the major site for synthesis of lipids. See, done. So main function of your lipids is, main function of your smooth endoplasmic reticulum is lipid synthesis. Tell me now in the chat quickly, what is the function of protein? What is the function of RER? What is the function of RER and what is the function of SER? In the chat, go. The function of RER is production of proteins. Smooth endoplasmic reticulum is releasing of lipids and steroidal hormones as well as steroidal hormones. In animal cells, lipid-like steroidal hormones are also synthesized in smooth endoplasmic reticulum. Done. Every single point covered. Quickly in the chat. Rough endoplasmic reticulum is protein. Smooth endoplasmic reticulum is lipids and steroidal hormones. Steroidal hormones like testosterone, progesterone, estrogen. Here only. Okay. Done. Yes, done. Endoplasmic reticulum, done and dusted. Quickly tell me the chat, any doubt here? Quickly tell me, any doubt here? This entire paragraph is decoded, right? Entire paragraph is decoded for you. So easy to understand. Very easy. Students, let me tell you honestly, botany is the easiest subject in the whole world. Botany is the easiest subject in the whole world. Learn the botany from me once and you will be able to solve every single question. It's only a matter of, see, you will learn the concept here. You will learn all the concepts here. It's only a matter of application, that's all. It's like how you apply it and how you solve questions. Keep on solving more questions and you will be so confident. Trust me, you will be super confident. So when I'm teaching the cell chapter here, go back home, sit aram se, open your DPPs, or do you want more DPPs? Tell me. Students, tell me if you want DPPs, I'll give you DPPs also. Do you want DPPs? After every class, tell me. After every class, if you want DPPs, I will upload at least 10 questions in my Telegram. After every class, if you want to do that also. Right? If you want every single day, after the class, based on the chapter we learn, on Telegram channel, I will upload at least 10 questions. On this NCERT Biology. In this Telegram channel, if you want from now onwards, we can make this a habit. Right? We can make this a complete habit. We'll make it a habit also throughout the year. We'll make it a habit. Okay. Whatever I teach here, In Telegram channel, if you go today only, I will upload some 10 to 15 questions, solve the questions and then sleep. Do not sleep before that. Only if you are able to do this, if you are serious, then only I will do it. If you are not serious, there is no point in me working hard. Okay, so because all this will take time and it will take my effort. So if you are serious, then only I'll do it. Okay, you want, right? Beautiful, amazing. So what I'll do is, students, whatever concept you learn with me after the class in the Telegram channel, I will upload some 10 questions, solve those questions and then sleep, go to sleep. Okay, done. I'm giving you some heavy promise I'm giving you. I'm giving you some heavy promise, so you keep up your bargain also. Study every single day. That is the only thing which I want in return. Okay. Students, this is your endoplasmic reticulum. Can you see? This is your endoplasmic reticulum. This is your nuclear membrane. From nuclear membrane, it is coming out. See, can you see? From nuclear membrane, your entire structure is coming out. Okay. Rough endoplasmic reticulum. Okay. This is your rough endoplasmic reticulum because ribosomes are here. Then we have smooth endoplasmic reticulum, no ribosomes. No ribosomes at all. Done. No ribosomes at all. Okay. No. Now we'll go to Golgi apparatus. Golgi apparatus, Amazon factory. This is going to be the Amazon factory. It is like Amazon factory. Okay. Very easy to understand. I will teach you one beautiful diagram. Okay. I'll show you one diagram. If you understand that diagram, this concept is laddu concept. Laddu concept. Understand now. Okay. First of all, who gave the term? 𝐂𝐤 Golgi in the year 1809 first observed the densely stained reticulate structure near nucleus. So tell me, what is the location? What is the location, students? Tell me. Location is going to be what? Location is going to be near nucleus. Simple. Near the nucleus. Okay. Now these were later named Golgi bodies after him. They consist of flat disc-shaped sac or cisternae. Okay. 0.5 micrometer to 1 micrometer diameter. These are stacked parallelly to each other. Can you see? They are stacked parallel to each other. They'll be stacked parallelly to each other like this. They'll be stacked parallelly to each other. Okay, parallel to each other. Varied number of cisternae are present in Golgi complex. Varied number of Golgi complex present. Okay. The Golgi cisternae are concentrically arranged near the nucleus. See, they are arranged near the nucleus with distinct concave. Understand now? There are two ends here. I want ultimate focus again. Ultimate focus. This is the diagram which I will be teaching you. Okay, ultimate focus. Understand. It's very easy to understand. Very simple. Okay. This is your Golgi. Can you see? It is parallelly stacked concentric rings. Parallelly stacked concentric rings. Yes. Parallelly stacked concentric rings. Concentric rings. Okay. Now understand this. There are two faces. Like everyone has two faces. In front of you there is something. In behind your back there is something. There are people, no, there are some negative people. In front of you they will say something. But behind your back, as soon as you turn, behind your back they will talk negative about you. Your Golgi complex is also like that. It has two faces. It has two faces here. Understand? It has something called as cis phase. It has something called as cis phase, also called as forming phase. Cis phase is also called as forming phase. Okay. Forming phase. And then we have trans phase. Then we also have something called as trans phase. Trans phase is also called as maturing phase. Is also called as maturing phase. Maturing phase. Okay. Trans phase. Now students, understand this very importantly. Diagram is very important. Okay. Now this is the nucleus. This right here is the nucleus. Okay. This right here is the nucleus. Okay. We have nuclear envelope here. We have nuclear envelope. From this nuclear envelope, we have endoplasmic reticulum. Okay. We have endoplasmic reticulum. Students, near this endoplasmic reticulum, there is a parallel stacked structures. There is a parallelly stacked structure called as Golgi complex. Golgi complex. This is your ER. This is your endoplasmic reticulum, ER. This is your nucleus. This is your nucleus. This is your nucleus. Now, students, understand this point very well. Understand this point very well. Okay. This ER, tell me, what does ER produce? Tell me, what does the ER produce? ER, endoplasmic reticulum, is producing proteins, right? It is producing proteins plus lipids. Yes, it is producing proteins plus lipids. Yes, it is producing proteins plus lipids. Okay. Now, whatever is produced by your ER, whatever is produced by ER, imagine it is producing here. Can you see? It is coming from here. ER is producing proteins plus lipids. That proteins plus lipid is taken up by, it is protein plus lipid is taken up by your Golgi complex. The protein plus lipid is taken up by Golgi complex, and this Golgi complex will make a separate layer around it. Can you see? This particular ER is producing some form of lipids or something. It is producing some form of lipids. This ER is producing some form of lipids, right? That lipids and proteins are taken up by Golgi complex, and they are done what? They are doing packaging. They are doing packaging here. Packaging done. Packaging done. So what is the main function here? The main function of your Golgi complex is packaging. Packaging of what? Products of ER. What products? Protein plus lipids. So we have protein and lipids. Protein and lipids from ER are completely packed by the Golgi complex. Completely packed by the Golgi complex. And understand this clearly. This phase right here. This phase right here is called as cis phase. Cis phase. Now focus here. From the cis, this is the cis phase. Cis phase is receiving what? The cis phase right here is incoming transport vesicles. Can you see? It is receiving vesicles. Yes, it is receiving vesicles. The vesicles are being completely packed. They are being completely packed. So as they are being packed, and it is moved out. It is going to be moved out later on. You see, it is being moved out. It is getting packed here, and it is moving out here, and it is moving out of the trans phase. Now this part here is called as trans phase. This is called as trans phase. Trans phase. This is called as forming phase or cis phase. Trans phase is called as maturing phase. Nucleus, endoplasmic reticulum. ER is producing some form of protein plus lipid. That protein plus lipid is taken up by your Golgi complex. It has been packed. It is how it is entering. It is entering from cis phase. Entering from cis phase, coming out of trans phase. It is moving out of trans phase. Is that point clear to all of you? Tell me in the chat. Is that point clear to all of you? Understand this. Understand the concept, your beautiful concept. Protein plus lipids are taken up by your ER through which phase? Through cis phase. Cis phase, it is entering, getting completely packaged out, and exiting from trans phase. This is the exit. This is the exit here. Trans phase. Exit. Yes? Okay? No. Beautiful. Understand the lines here? Constantly arranged near the nucleus with a distinct convex cis or forming phase, also called as convex. Convex cis forming phase. This is convex. Convex forming phase, and concave trans phase or the maturing phase. I told you, maturing phase. The cis and the trans phase of the organelles are entirely different but interconnected. They are different but they are interconnected. Different but interconnected. Okay. The Golgi apparatus principally performs the function of packaging, Amazon factory, packaging material, to be delivered either to the intracellular target or secreted outside the cell. Outside the cell or inside the cell. Okay. Materials to be packaged in the form of vesicle from the ER. I told you, it comes from the ER. ER fused with the cis phase. See, it is fusing with the cis phase first. cis phase of Golgi apparatus and move towards the maturing phase. So it is coming towards the cis phase and it is traveling towards the trans phase or the maturing phase. This explains why Golgi apparatus remains in the close association with endoplasmic reticulum. That is why it is in close association with endoplasmic reticulum. Okay. Now, a number of proteins and synthesized by the ribosomes on the endoplasmic reticulum are modified. This modification is called as glycosylation. It is called as glycosylation. What is the meaning of glycosylation? The main meaning of glycosylation is addition of carbohydrate. Okay, understand this. A number of proteins synthesized by the ribosomes on the endoplasmic reticulum are modified in the cisternae of Golgi apparatus before they are released from its trans phase. So protein is entering, glycosylation, glycoprotein. Lipid is entering, glycosylation, glycolipid. You understand? Protein synthesized by ER is getting converted to glycoprotein. Lipid, glycolipid. Glyco is sugar. Carbohydrate attachment. Golgi apparatus is an important site for formation of glycoprotein and glycolipid. Glycosylation is happening. Glyco is addition of carbohydrates. Understood? Concept. Brain, it went little bit. 10% it went. 10% also it went. If I'll be happy. If it went 10% also, I'll be happy. Okay. If you did not understand, re-watch it again. I've explained beautifully. Concept should be clear. Nice. That is why it's called as glycoprotein, glycolipid. This protein is coming from ER. This protein is coming from where? This protein is coming from RER, rough endoplasmic reticulum. Lipid is coming from smooth endoplasmic reticulum. What is the function of Golgi body? Golgi body is just adding the glyco part. It is adding the glyco part. It is adding the glyco part. Understood, no? 7 p.m. right? Okay. 7 p.m. I'll give them a break. 7 p.m. I'll give them a break. Don't worry. Okay. Now lysosome students. Now next concept is lysosomes. Ready? Not break now. Oh my god. Not break now. Break at 7 o'clock. Because Shreyas sir is coming and giving an announcement. 7 o'clock is the break, not now. Okay? Don't worry. Now, lysosomes now. Now, these are membrane-bound vesicular structures. Membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. So, if I ask you a question, how is the lysosomes coming out? It is a product of your Golgi complex. Lysosomes is the product of your Golgi complex. Remember that. Okay? Okay. Yes. The isolated lysosomal vesicles has been found in a very rich in almost all types of hydrolytic enzymes. So if you ask me what is present inside the lysosomes, we have hydrolytic enzymes. Can anyone tell me what are these hydrolytic enzymes? What are these hydrolytic enzymes, students? Hydrolytic enzymes are enzymes which are able to break bond with addition of water. They'll add water, break the bond. Hydrolytic. Hydro means water, lytic means break. They're breaking the bonds with the help of water. Examples are very simple. Lipases, proteases, carbohydrases. Lipases will break lipids, proteins, carbohydrates. It will completely break it out. Optimally active at acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acids as well as nucleic acids. See here students, this is your nucleus, nuclear membrane, endoplasmic reticulum. The product of your endoplasmic reticulum is taken up by Golgi complex. This is your, tell me which phase is this, tell me students. This is your cis phase, this is your trans phase. Cis phase it is entering, trans phase it is coming out. Then we have the lysosomes. Lysosomes. Understood? Lysosomes. No, students. And your lysosomes are also called as the suicidal bag. Not written here. They are called as suicidal bags because every single time there is something wrong with the cell, it will kill the cell. It will already kill the cell. Okay. Now, students, next we understand vacuoles. Can we start, students? Can we start the vacuoles now? Can we start the vacuoles, students? Can we start the vacuoles? Quickly smash the like button. Tell me, it's very interesting, no? Very interesting to learn about the cell. Every single line of NCERT we are covering and going without any breaks, right? Every single line. Yes, we can start. Amazing. Amazing. Divya is talking to someone randomly. One nice conversation is going with Divya. Divya is talking to herself. Nice, nice. Okay. Now we start with vacuoles. The vacuoles in the membrane bound space found in the cytoplasm. They are found in the cytoplasm. Vacuoles are found in the cytoplasm. Understood. Now students, tell me, where do you find large vacuoles in the case of plants or animals? Tell me, where do you find large vacuoles in the case of plants or animals? Quickly, in the go, chat. We find in the case of plants. In the case of plants, we find large vacuoles. Okay, more storage. It contains water sap is present inside. Okay, excretory products present inside. Yes, and other materials not useful for the cell. Basically, this is a dustbin. It is somewhat of a dustbin. Okay, whatever is not required will be thrown there. Nicely you throw there. Okay, it is like a bed also. Whenever you come, your bed will always have clothes. No, bed will always have some clothes lying. Some towel will be on the bed. Keep throwing on the bed everything here, or dustbin. Okay. The vacuole is bound by a single membrane called as tonoplast. Very important. Very important. The membrane of vacuole is called as tonoplast. It is called as tonoplast. In plant cells, the vacuoles occupy the 90% of the volume of the cell. Large cell, no, large vacuole plant cell, 90%. In plants, tonoplast facilitates the transport of numerous ions and other materials against the concentration gradient. Understand this. In the case of plant cell, all the ions, students. In the case of plant cell, all the ions from the low concentration, they are going inside the vacuole against the concentration gradient. See, usually what happens? Transportation is always easier from high to low. From higher concentration to lower concentration, always it is very easy to go. Just like your marks. Tell me, if you're easily scoring around 650 marks in mock test, you don't study for 5 months or 6 months, you don't study anything, you don't revise anything. Easily you will come to 200 marks. From 650 you will come to 200 easily. This is very easy. Don't study, it will happen. But from going from 200 marks to 650 marks, this is more difficult. This is more difficult, right? This is more difficult. You have to work hard. You have to work very hard, right? But here also, against the concentration gradient, ions are sent into the vacuoles, inside the vacuoles. Okay, that is what they're telling you. That's what they're telling you here. Okay, in plants, tonoplast facilitates transport of numerous ions and other minerals against the concentration gradient into the vacuole. Hence, their concentration, ion concentration, is slightly higher in the vacuole than in the cytoplasm. Can you see? There is inside the vacuoles, there is high concentration of ions compared to the cytoplasm. Now, in amoeba, contractile vacuole is there. It can change shape. It can change shape and take the food inside. Contractile vacuole is important for osmoregulation and excretion. Osmoregulation as well as excretion. Okay. Now, in many cells, as in protists, food vacuoles are formed by engulfing the food particles, vacuoles. So vacuoles are important sometimes. See, this is your amoeba. It has contractile vacuole here. With the help of contractile vacuole, it can eat. Done. and also excrete food. Osmoregulation can also be done. Osmoregulation can also be done. Mostly it is osmoregulation, excretion. Food, there is food vacuole. Food, there is food vacuole. Contractile vacuole is usually excretion as well as osmoregulation. Understood? 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 Yes or no? Quickly in the chat. That is osmoregulation. Contractile vacuole, osmoregulation as well as excretion. Food vacuole for food particles, to eating the food particles. Now students, now let's start mitochondria. Students, can we start mitochondria after 7:10 or 7:10? We will take on 20 minutes break now. Aram se chill. Okay. Shreyas sir will be coming live, and he'll be giving an amazing surprise. It's an amazing surprise for all of you. So can we start? Because if I start mitochondria right now, it will not get over in 10 minutes. If I start mitochondria right now, it will not get over in 10 minutes. It will take more time. Okay. So can we start mitochondria after Shreyas' announcement, and then once Shreyas sir is done, can we start with mitochondria again? Can we do that? Okay. All of you also need some break. No. Go drink some water. Go eat something. Go and come. Go eat some biscuit and come. Go run. Go grab some biscuit or try some chips or get on dinner. Also get off. Start eating the dinner also. Okay. So once Shreyas sir finishes, then we'll start. So but promise me you'll come back. That is very important. Promise me all of you will come back, because mitochondria chloroplast, many questions will come. Mitochondrial chloroplast, many questions will come, and it is going to be very important for you. Okay. So come back, and it is a very good surprise. It's going to be such a good surprise. All of you will be shocked. Like, oh my God, what is happening? You'll tell. Okay. So come back later on. We will start with mitochondria later on. Okay. Let's give a small break right now, because if I start now, it will not get over, obviously. Okay. Now students, can I ask you some questions till now? So before Shreyas starts, can I ask you some questions? Why to waste time? Okay. Tell me, what is the layer of vacuole called as? Quickly in the chat. What do we call the layer of vacuole? Quickly. I want rapid fire now. Rapid fire questions. Quickly. The layer of vacuole is called as? It's called as tonoplast. Nice. It is called as tonoplast. Nice. Okay. How does the endoplasmic reticulum divide? How does the endoplasmic reticulum divide the entire cell? There are two types. No, entire cell can be divided into two parts with the help of ER. What do you call that? Correct. It is called as luminal and extraluminal. Luminal as well as extraluminal. Now, tell me quickly in the chat, what are the organelles under endomembrane system? Quickly go, all four of them. What are the organelles under endomembrane system? First one is endoplasmic reticulum. Second one is Golgi complex. Third one is lysosomes and vacuoles. Lysosomes as well as vacuoles. Nice. Now, your Golgi complex has two phases. What are the two phases now? Tell me. What are the two phases in the Golgi complex? One is called as a cis phase. Okay. The cis phase is also called as, what is the other name of cis phase? Quickly tell me in the chat. What is the other name of cis phase? Correct. Cis phase is also called as forming phase. It can also be, it is the convex structure. Okay. The other phase is called as trans phase. Trans phase is also called as maturing phase. Now question is, what are the products of your rough ER? What are the products of your rough ER? Quickly go in the chat. Rapid fire now. What is the products of your rough ER? Protein synthesis. Nice. What is the product of your smooth ER? What are the products of your smooth ER? Go quickly in the chat. Products of your smooth ER. Correct. The smooth ER produces lipids as well as your steroidal hormones. Give me an example of steroidal hormone. Example of steroidal hormone. Correct. Progesterone, testosterone, estrogen. Nice. Correct. Now tell me, products of your ER is taken up by Golgi.
by which phase? The products of your ER is taken up by Golgi complex by which phase? Cis phase or trans phase? Cis phase or trans phase? Quickly go in the chat. Quickly go in the chat. Which phase? Cis phase or trans phase? Cis phase. Nice. Cis phase. Cis phase it is entering. Trans phase it is coming out. Cis phase it is entering. Trans phase. Madhumita. No. Cis phase it is entering. Always remember. Cis phase it is entering. Always remember. Cis phase it is entering. All the products of your ER. All the products of your ER is entering from cis phase. It is exiting from trans phase. It is exiting from trans phase. Remember that. Remember that, all of you. Remember that. Okay. One more question. No? Nice. We can see, students, remember this. Questions we can always keep on making. Questions we can keep on making questions. There's no stoppage for that. Now, students, tell me, what are the products of your Golgi complex? What are the products of your Golgi complex? Products of your Golgi complex. Quickly go in the chat. Glycoprotein as well as glycolipids. Nice. Glycoproteins as well as glycolipids. Okay. Nice. Glycoproteins as well as glycolipids. Okay. Now, in the case of amoeba, which type of vacuole do you find? In the case of amoeba, which type of vacuole do you find? In the case of amoeba, which type of vacuole do you find? In the case of amoeba, we find something called as contractile vacuole. Nice, beautiful. Contractile vacuole. Contractile vacuole. Here, contractile vacuole. In the case of amoeba, tell me the function of contractile vacuole. What is the function of contractile vacuole? What is the function of contractile vacuole? Function is very simple. The function is going to be osmoregulation as well as excretion. Osmoregulation as well as excretion. Nice. Now, lysosomes is here only. I can't ask you. Students, lysosomes has which type of enzymes? Lysosomes has which type of enzymes? Which type of enzymes do you find in lysosomes? Hydrolytic enzymes. Nice. Hydrolytic enzymes. Do you realize we summarized the entire part we did today with the help of questions? Did you like it? If you come back, trust me, I will do the similar type of exercise by the end of the class. An entire chapter is being revised in your mind. Entire chapter is being revised in your mind. That shows you the concept learning process. Okay. Now I'll ask you one more question. What do you call the connections between the two cytoplasms? What do you call the connection which is found between the two cytoplasms? Two cytoplasms. Go in the chat. It is called as plasmodesmatal connection. Plasmodesmata. It is called as plasmodesmata connection. What about between the two cells, between the two plant cell, we find something called as some glue. What do you call that glue? Between the two cell walls, we find some glue which holds the two cells. What do you call that? That is middle lamella. Okay, that is middle lamella. Right? Did sir make it to 6:30? Don't tell me. 7:30, huh? Sir made it to 7:30. Ayyo. Okay, students. Your revision is also done. Can we start with mitochondria then? Can we start with mitochondria now? Can we just can you even check, is the surprise postponed to 7:30? Can you even check in the YouTube? I don't know. Sure, it is not showing for me. Just check once. If it is 7:30, we'll start mitochondria. We will start with mitochondria then. What is the hurry? Okay. You want more questions, huh? Do you want more questions after we do the mitochondria and chloroplast? After we finish mitochondria and chloroplast, more questions. How was the question round? Do you realize all these questions were made on spot? All of these questions were made on spot, rapid fire questions on spot. And you were able to answer. You were able to answer all of them. Okay. It is postponed. Nice. Good for you only. No. Good for you. Will be finished mitochondria. If that is the case, we'll finish mitochondria then. Okay. Last question only. I'll ask you last question.
Last two questions. Okay. What is middle lamella made up of? What is middle lamella made up of? Go. What is middle lamellae made up of? Middle lamellae made up of. Quickly go in the chat. Break, wait, na. Wait, hurry, I can't see that. Hurry, shine, wait. Middle lamellae made up of. I know you didn't start your break. Luckily, we didn't start the break. We can teach mitochondria now. What is middle lamellae made up of? Calcium pectate. Nice. Calcium pectate. Okay. Calcium pectate. Nice. What is the cell wall made up of in the case of algae? That is the last question. Last question is what is the cell wall made up of? Cell wall is made up of in algae. Go. I saw, sir. I saw 7:30. Students only told me. Students only told me 7:30 announcement, so I can continue. Nice. Now, students, let's start with mitochondria. Okay. Quickly in the chat, what is the other name of mitochondria? What is the other name of mitochondria? Quickly go in the chat. What is the other name of mitochondria? Mitochondria is also called as the powerhouse of the cell. It is called as powerhouse of the cell. Why? Why is it called power of the cell? Because it is a site for cellular respiration. It is a site where we produce ATP, yes, adenosine triphosphate. That is why it is called as powerhouse of the cell. Yes, it is called as powerhouse of the cell. Okay. It is unless specifically stained are not easily visible under the microscope. So you cannot see them under the microscope. You cannot see them under the microscope usually. Why? Because they are very tiny, they are very small. They are very small. Okay. The number of mitochondria per cell is variable depending upon the physiological activity of the cell. For example, again, heart cell. Tell me, heart cell and cheek cell, which one needs more mitochondria? Go in the chat. Which one needs more mitochondria? Which one needs more mitochondria? Cheek cell, because you're getting slapped. Tell me, which one has more mitochondria? Heart cell. Cardiac cells has more mitochondria because more requirement. Cheek cell, nothing. It is just lying there. Doesn't require mitochondria, right? So it varies completely. Okay. In terms of shape and size also, considerable degree of variability is observed. Okay. Typically is sausage shaped or cylindrical, having diameter of 0.2 to 1 micrometer, average length that is 1 to 0.4 micrometer in length. Each mitochondria is double membrane. Students, here is a mitochondria. Each mitochondria, can you see? Each mitochondria is double membrane. Can you see, students? It is going to be double membrane structure. Can we draw here? Yes, we can draw. It is going to be sausage shape. It is going to be sausage shape. It is going to be somewhat sausage shape. Yes. It is going to be somewhat sausage shape. And it is double membrane. It is going to be double membrane structure. It is going to be double membrane structure, students. Here we have something called as the outer membrane. We have the outer membrane and we have the inner membrane. We have the inner membrane here. Outer membrane as well as the inner membrane, students. Outer membrane as well as the inner membranes. Now inside we have some type of foldings. Inside the mitochondria there are some types of foldings, students. There are some types of foldings inside the mitochondria. Such foldings are called as cristae. We call them cristae. Here, can you see? They are called as the cristae. Now, what could be the main function of cristae? Tell me, students. What could be the main function of cristae? The main function of cristae is to increase the surface area. Increasing the surface area is going to be the main function here. Main function. And students, remember the outer membrane here. The outer membrane here is much more permeable. It is much more permeable. Outer membrane is much more permeable compared to the inner membrane. Compared to the inner membrane. Okay. Inner membrane. Clear? Inner membrane, we can say it is selectively permeable. It is selectively permeable. Selectively permeable.
selectively permeable. That is the inner membrane. Inner membrane. Now, students, tell me, do you find the genetic material here? Do you find the genetic material? Tell me. Tell me, students. Do you find genetic material here? Quickly tell me in the chat. Do you find its own genetic material here? A single double-stranded DNA can be found here. A single, students, remember, a single double-stranded DNA can be found here. Yes. Single double-stranded DNA can be found here. Yes. Now, does it have ribosomes? Does it have ribosomes? Yes, sir. It has ribosomes here. It has ribosomes here. That is 70S ribosome. 70S ribosome, not 80S like your cytoplasm. Cytoplasm is 80S, but your endoplasmic reticulum has 70S ribosome. 70S ribosome. Now, students, on this cristae right here, we find certain bat-like structures. On this cristae, we find certain bat-like structures. Can you see? Bat-like structures. These bat-like structures are nothing but elementary particles. They are called as elementary particles. Elementary particles. The other name for the elementary particles is oxysomes. We can also call them oxosomes. Oxysomes. We can also call them F0, F1 particle. F0, F1 particle. They can also be called as ATP synthase. ATP synthase. We can also call them ATP synthase. Now, students, this is crystalline A. Then what are this, students? This part here. This part right here is the matrix. It's going to be matrix. It's going to be matrix here. It's going to be matrix. Then this is going to be matrix right here. Matrix. Okay? Matrix. This is the structure of your ATP synthase. Structure of your ATP synthase. See here. Membrane bound structures with outer membrane and inner membrane dividing its lumen distinctively. Dividing its lumen distinctively into two aqueous compartments. The outer compartment and the inner compartment. The inner compartment is filled with dense homogeneous substance called as the matrix. We decided that was the matrix. Okay, matrix. The outer membrane forms the continuous limiting boundary of organelle. The inner membrane forms numerous infoldings called as the cristae. So, from the inner membrane we have the cristae. From the inner membrane we have the cristae. Towards the matrix, the cristae increases the surface area. The two membranes have their own specific enzyme associated with the mitochondrial functions. So, students, in the matrix, all the enzymes are present. In the matrix where the Krebs cycle takes place, yes, Krebs cycle takes place there, all the enzymes are present except one enzyme. What is that enzyme? Very good question. In the matrix, all the enzymes are present except one enzyme. Which is that one enzyme which is not present in the matrix? They produce cellular energy in the form of ATP by power of the cell. That's why the powerhouse of the cell. The matrix also possesses single circular DNA. It's double stranded. Always remember it is double stranded. And a few RNA molecules. Ribosomes 70S. The components required for synthesis for protein. The mitochondria divides by fission. Very important line. Mitochondria divides by fission, just like bacteria. Just like bacteria, it divides by fission. Bacteria divides by fission. Any doubt here? Quickly ask me. Any doubt here? We finished the entire paragraph. Entire paragraph is done. Entire paragraph is done. Any doubt here? Quickly ask me. Any doubt here, students? Any doubt here? Quickly ask me. Yes, Harishri, they are like fathers. Why? Because there is something called as endosymbiosis theory. Endosymbiosis theory says that multiple prokaryotic organisms were engulfed by one big organism, and we got eukaryotic. That's why they have 70 years. That's why they have the 70 years. Any doubt? Quickly ask me, students. Any doubt? There are one enzyme, missing enzyme is called as succinate dehydrogenase. Succinate dehydrogenase. It's called as succinate dehydrogenase because succinate dehydrogenase is part of your electron transport chain. Okay, remember that. Remember that. Remember that. Okay. Succinyl dehydrogenase. Harish, okay, very difficult. Names very difficult for me, students. Names is one thing which I am not able to master. I don't know why. See, I can't even see. Savita, I can say. Jenlisa, Pavitra, Rishmita, Harshin, right? Harshin, Tanuja, Divya, Madhumita, Rashmika. See, some names are very difficult. Some names are very difficult. Sir, ribosomes in eukaryotic plasmosome, sir. Ribosomes in the, I'll tell you, it's coming up anyway. It's 80 s. In the cytoplasm, it's 80 s. In the chloroplast, as well as mitochondria, it is 70 s. It is 70 s. Okay. Shin. Okay, Shin, sir. Harshin or Harshine. I give up. Names, I will never. You will only scold me later on, sir. What are you telling? Okay. See, Rudranath Upadhyay. Next, I can't tell. Rudranath Upadhyay is very easy to tell. Okay. Cool. So, that is done. Now, students, can we do plastids now? Can we do plastids? Okay, outer membrane plastid, we can do this. And can do this in next class, actually. Students, actually, after plastids, we'll stop the class today. What's it? Let's not drag the class too long. Let's not drag the class too long. After plastids, we'll stop the class today. I think we've been almost 2 hours. It will become almost 2-hour class. We'll stop there. Okay. In part 3, because that requires, see, some amount of brain cells are used now. Some amount of brain cells are used. So you can't, take so much of the brain cells also, because to learn flagella structure, to learn the structure of cilia and learn the structure of nucleus, you require fresh brain cells. Right? So after plastids, we'll stop the class. Let's Shreyas do it peacefully. Otherwise, Shreyas will be like, oh, and all. Okay. So we'll start. Now let's do plastids. Very easy to understand. Plastids. Okay. Plastids are found in all plants and in euglenoids. Now, if I give you a question, if I give you a statement, right? If I give you a statement, plastids are only found in plants. True or false? Tell me, true or false? Plastids are only found in plants and no other organisms. True or false? Only found in plants. It's going to be false. Because plastids can also be found in the case of euglenoid, which is a type of protist. Yes, type of protist also can be found. These are easily observable under the microscope as they are very large. Okay. They bear some specific pigments, thus imparting specific colors to the plants. Okay. They can produce colors. Based on the type of pigment, plastid can be classified into chloroplast, chromoplast and leucoplast. Chloroplast gives which color? Chloroplast gives green color. Chromoplast produces red, yellow, orange. Leucoplast is colorless, usually colorless. Don't tell white and all. Okay. The chloroplast contains chlorophyll and carotenoids pigment, which are responsible for trapping the light energy essential for photosynthesis. That is the function. In like chromoplast, fat soluble carotenoids pigments like carotenes. See, in the case of chromoplast, we have carotenes, xanthophylls and other pigments are present. Done. Okay, then. Now, this gives the part of the plant the yellow, orange, red color. See, yellow, orange, red color because of the chromoplast. Now, last one is
leucoplast. Transparent, sir. It is basically colorless. Yeah, transparent only. See, not transparent. It doesn't have any pigment which gives color basically. See, pigment doesn't have to give color always. Remember that. Pigment are chemical substances which help in making the light energy excited. That is the main function of pigments. Okay. The leucoplasts are colorless. See, colorless plastids of various shape and sizes. But main function of leucoplast is storage. Main function of leucoplast is storage. Very important point now coming up. Amyloplast stores carbohydrates, amylopectin, remember it like that, amylopectin and amylose, starch. Elioplast, that stores oil and fats, whereas aileroplast stores protein. Aileroplast, remember students, aileron layer in the case of seed, aileron layer is a proteinaceous layer. We learned somewhere, right? In the seed we learn aileron layer is a proteinaceous layer. So it stores protein. So remaining is going to be fats and oil is aeroplast. Elio sounds like oil, elio oil. So it's going to be oils and fats. Understood? Understood, students? That's how you can remember also. That's how you can remember. That's how you can remember it. Okay. Amylopectin, starch. Elio, oil. So stores oil. Aileron layer is a proteinaceous layer. So you can remember it, protein. Done. Seed. Remember it, seed. Now, this is the structure of a chloroplast. Again, it is a double membrane. Again, it is a double membrane structure. We have the outer membrane and we have the inner membrane. Outer membrane as well as the inner membrane. Two different layers are here. Two different layers are here. Outer membrane as well as inner membrane. Now here, tell me in the chat, here we have this coin-like structures. We call them thylakoids. Thylakoids. Here we have the, let me draw the structure, students. Let me draw the structure. Let me draw the structure. Here we have the two layers. Here we have certain coin-like structure. This is the outer layer. Outer layer. This is the inner layer. This is the inner layer. This is the outer layer. Okay, outer layer, inner layer. Here we have certain coin-like structure. They are called as thylakoids. They are called as thylakoids. The entire stack is called as grana. It's called as grana. Here also we have thylakoids. Thylakoids. Between the two thylakoids, students, between the two thylakoids there is a connection. There is a connection. The connection between two thylakoids is called as your, it's called as your stromal lamellae. Stroma lamellae. Stroma lamellae or lemma. Stroma lemma or stroma lamellae. Okay. Now tell me, where does the light reaction happen and where does the dark reaction happen? This part is called as the matrix. This is the matrix. Stroma. Stroma. Also a matrix. Remember, students, light reaction always happens in the thylakoids. Here we have the light reaction in both the places. Even in the stroma lamellae as well as the thylakoid, light reaction takes place. Dark reaction happens in the stroma. Dark reaction takes place inside the stroma. Remember that. Remember that, all of you. Okay. Dark reaction takes place there. Now, students, majority of chloroplasts of the green plants are found in mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-shaped organelles having variable length. Their number varies from 1 per cell in Chlamydomonas, only one chloroplast, or even many can be there, or 20 to 40 per cell in mesophyll cells. Like mitochondria, chloroplasts are double membrane structures of two inner and outer layer. Relative permeable, yes, is relatively permeable. Out of the two, inner chloroplast membrane is, inner one is relatively less permeable. The space limited by the inner membrane of the chloroplast is called as stroma. Inside part is called as the stroma. A number of organized flat structures is called as thylakoid are present in the stroma. Thylakoid are arranged in a stack called as
grana. Yes, entire stack is called as your grana. Yes, integral thylakoid. In addition, there are flat membrane structure called as stromal lamellae connecting the two thylakoids. Stromal lamellae between two thylakoids. The members of thylakoids encloses a space called as your lumen. Lumen is nothing but inside the thylakoid. For example, students, this is a thylakoid, right? These are thylakoids. Inside the thylakoid, this part inside part is called as lumen. Inside part is called as a lumen. Okay. The stroma of chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. Stroma, that is for dark reactions. Rubisco enzyme they're talking about here. Okay. It also contains small double-stranded DNA. See, it has own DNA. It has own DNA, even ribosome, 70S ribosome. And ribosome. Which ribosome? 70S ribosome. 70S ribosome. Chlorophyll pigments are present on thylakoid. Ribosomes of the chloroplast are smaller, 70S, than the cytoplasmic ribosome, which are 80S. Done. Done. Students, with that, today's class is over. Before I go, students, I think I have told all of you almost all of you. I will repeat again if I have not told you. Students. If you want to be part of Vedantu's paid batches and online or offline both, but you cannot afford it. Sometimes money is a major constraint, right? If you cannot afford it, we are giving, if you are smart enough, if you are on merit, if you are smart enough and if you are educated and if you are able to work hard, we are giving up to 100% scholarship. Up to 100% scholarship. Okay. When I say up to, it means up, not complete 100%, up to 100% scholarship. The exam is going to be MVSAT exam, Mega Vedantu Scholarship Admission Test. You can get up to 100% scholarship on Vedantu's paid classes. And this examination is free of cost examination. All you need to do is apply it. If you go to the description of this video, you will find a link. Quickly register yourself, and then write the examination. Show us your worth, and take home, scholarship. If not scholarship, you can get some things like PS5, iPad, iPhone, drones, even the entrepreneurship classes by our CEOs. CEO will sit with you. Vedantu CEO will sit with you and talk to you. Okay, it's matter of your hard work. That's all, and it is completely optional. But I would recommend you to do it because it is free. You have, as I always say, this examination, you have nothing to lose, everything to gain. So write it. Okay. So with that being said, today's class is done. Students, commence, because that's how the video will get ahead. Students will get to know that at Vedantu we have a teacher called Baswaraj sir who will teach you line of NCERT, who will make you fall in love with botany. There's a teacher like that. So the only way students will get to know that yes, there's a teacher like this is by your comments, right? So students, let me know in the comment section how was today's class and how was the teaching, and if you love botany. Okay. And botany is super easy. Trust me, botany is going to be super easy. So easy it is. Was there any difficulty today's class? There was no difficulty at all. It has been super easy with all of you. Okay. So have an amazing day ahead. Thank you so much, students, for attending today's class. And I will see you in the part 3. Where we'll be teaching you the remaining part. Remaining part is very simple. Can you see? We have the entire, we basically have the nucleus, structure of chromosomes, structure of nucleus, centrioles, structure of your flagella, and we have the ribosomes. That's all. Maximum the class will be what? 45 minutes or 1 hour. Next 1 hour class will be there. That's all. And if you want, I will solve some questions also. Okay. So take care students, and bye-bye. I'll see you in the next class. Take care all of you. Bye-bye. Have a great day ahead. Bye-bye.